And we're live. Walter Isaacson, thank you so much for doing this.
Hey, Andrew, great to be with you, sir.
So, we're going to talk about your book, Codebreaker, which came out in 2021. And the book focuses on the discovery of the CRISPR gene editing system, and in particular, the role played by Professor Jennifer Doudna. You've written about political figures, artists, entrepreneurs, and others. What made you want to write about a scientist and a scientific topic?
Ever since I was at Time magazine and we were picking persons of the year, I realized that scientists, engineers, technologists, and people who went into the business of innovation affected our lives more than those politicians we often wrote about. And so, pretty early on in my career, I started writing about people like Einstein or Steve Jobs, who I think affected our lives a lot more than whoever was the Secretary of State or even the president. And in this case, Einstein brought us into the era of physics in 1900 or so. That leaves everything from the nuclear bomb to GPS to lasers, all coming out of his Miracle Year papers of 1905. Likewise, you have a revolution that's a digital revolution that comes with the invention of the computer, the microchip, and the internet. And Steve Jobs and Bill Gates, both people I've written about at times, they lead that revolution to make it personal, into personal computers. I think around the year 2000, we entered a new revolution, and that's a life sciences and gene editing revolution. And around the year 2000, we sequenced the human genome. We were able to figure out all those letters of a code of DNA and what type of genes they might express. And what happens with CRISPR and Jennifer Doudna is they come up with a tool to edit DNA. And I think that will help shape the next 50 years of history.
I'm excited to go into a lot of different topics related to this book, but I want to just first set the stage. So, in the late 90s, early 2000s, what did scientists begin noticing about bacterial DNA?
Bacterial DNA has a repeated set of sequences, some of them do. Now, bacteria don't have a lot of DNA, so why would they have all these clustered, repeated sequences that say the same thing? And they named those things CRISPR, meaning the scientists who discovered them. They said, how come? How come bacteria do this? Well, it turns out, if you think, Andrew, that you and me and the rest of humanity has problem with viruses, bacteria really has problem with viruses. That's the biggest battle on this planet for the past 2 billion years is bacteria trying to fend off viruses. And it turns out that CRISPR, these repeated sequences, is a way for bacteria to remember, to take a mugshot of a virus that's attacked them and then find a tool that allows them to chop the DNA at that sequence if a virus has that sequence and that attacks them. Now, this might not seem all that amazing until we discovered about the same time that we're getting hit by a massive virus attack, the coronavirus. And likewise, when we discover we can take that tool and use it to edit our own DNA.
Right. And so around, let's say 2010, Jennifer Doudna, she begins to understand what some scientists who had sort of previously discovered around there are these repeated sequences in bacterial DNA. And what does she do with that information?
Well, first of all, she figures out how to turn it into a tool so that we can figure out spots in our own DNA and we can guide a pair of scissors to that spot. By pair of scissors, technically, I mean an enzyme, which is a protein that knows how to cut things. And so she creates a tool called CRISPR-Cas9, which basically says if you want to cut DNA at a particular sequence, you just program it in and you put this enzyme and you connect it to it, and that tool will do it. A lot of people were working on it, but she, along with her research partner, Emmanuel Charpentier, and a lot of other colleagues, were the first to show how you turn this bacterial weirdness, that bacteria have these repeated sequences, and you say, I can make a tool out of it.
So her team, they initially show that they can do it in a bacterial cell. And upon releasing that paper in 2012, a race then begins to see which team of scientists around the world can do the same thing, but in a human cell. I don't want to spoil the book too much, but that race is sort of a big focus of the book. And I think that leads me into this topic of scientific credit. So ultimately, the discovery and the translational work here is due to the work of a lot of different types of researchers around the world. What do you think the story says about scientific discovery in our modern world, where breakthroughs or elements of breakthroughs can be shared instantly across the world?
I think it's important to understand that innovation is a team sport, that creativity is a collaborative effort. And that's what the story of CRISPR shows. But it's also, as you just mentioned, a competitive effort. You get teams of people, and they get rewarded if they're the ones who get there first. For example, there's still, as you sort of hinted at, a patent dispute going on between Jennifer and her team versus a team that's at MIT and Harvard that Eric Lander and Feng Zhang and some others lead. Now, you don't have to worry about a spoiler alert because just this past week, the judge sent it back for more evidence and more trial. So it's still unsettled. But one of the things that causes us to compete and not share information too much is we want to be the ones who get the patent. And that seems bad. On the other hand, what's driving these people to stay up all night and work weekends is they want to get the patent. So, whatever your listeners, whatever they do in life, whether they're venture capitalists in private equity or they're baseball players or they're people making toothpaste, you know that both competition causes people not to collaborate, but it does cause them to really speed up innovation. And that's what this book is about. How does innovation occur and what drives it? There are other factors, of course, besides patents. You want to win the Nobel Prize. Jennifer ended up winning that one with Emmanuel Charpentier. You want to be the first to publish. You want the esteem of your colleagues. So we have a lot of things that drive us both to compete in a race, but also to collaborate with other people.
After writing this book, how difficult do you think it is to sort of adjudicate and accurately allocate the credit of a specific scientific breakthrough?
That's always been difficult. And take one of the greatest technological breakthroughs of our era, and that's the invention of the microchip. You have Texas Instruments doing it with Jack Kilby. You, of course, have the famous team at Intel with Robert Noyce, Gordon Moore doing it, and eventually Andy Grove. They do it at about the same month. They come up with ways to make a lot of transistors on a piece of silicon and call it a microchip. How do you adjudicate the credit? At one point, after years of a patent battle, Bob Noyce calls up Jack Kilby, his rival on the microchip, and says, let's quit fighting over divvying up the proceeds until we finish robbing the stagecoach. Let's just agree we all invented it. In my book, I try to figure out the same with CRISPR. Someday, I hope all the sides will say, okay, we all deserve credit. But if you sit in a meeting, Andrew, with a variety of people, and there's a good idea that emerges from that meeting, and 6 months later it makes $1 million, you can have everybody in that meeting thinking, hey, I was the one who deserves most of the credit for the meeting. So we do that in our daily lives. I think in science, we actually do it better. The patent laws and the patent courts, believe it or not, they're damn good. It may be complicated, but they're understanding things like CRISPR and who got it to work first, who reduced it to practice, as it's called, meaning you made it work. Likewise, we have a system of publications and peer-reviewed journals. And if you beat somebody by 24 hours getting something published, then you put a stake in the ground. You were the first to publish it. So we have ways of adjudicating credit. And I must say, people criticize all these fights over things. I think especially in America, but even around the world, scientists and technologists are the best even if it involves patent battles that go on for a while. They're the best at saying, here's the rules of the road for who gets credit. Starts in whatever, 1790, when in the Constitution of the United States, they put in that clause that Congress shall have the right to grant patents and copyrights. And we developed a system to say who gets the patents, who gets the copyrights.
That's encouraging to hear. So I want to ask about Jennifer Doudna. You have spent time covering and writing about a lot of different types of very successful figures. How is Jennifer similar to the entrepreneurs you've spent time with and written about, and how is she different?
One way that she's different is, excuse my language, but she's not as much of a jerk. The technical term begins with an A. I don't know if you use it on podcasts.
We do.
But if you look at Steve Jobs, you look at Elon Musk, frankly, if you look at Jeff Bezos in the early days of Amazon or Bill Gates in the early days of Microsoft, they could be assholes. And they were really rough on people. And one of the things I try to write about is, do you have to be so rough in order to get results? Well, Jennifer, like Benjamin Franklin, is a case study of somebody whose talent was not driving people mad and insane and being rough on them and firing, but being a team builder, nurturing people, bringing them together. And there's no right answer. If we had only team builders and nurturers in our society, we probably wouldn't break enough China to have innovation. If we only had people breaking China and calling each other assholes and firing people, we probably never would get the microchips out the door, the CRISPR into the pharmacy. So I think you need both. And that's why people like me write biographies as opposed to books that say 7 Secrets to Leadership or the 12 Paths to Innovation. It's like you got to understand people. You got to read Shakespeare, you got to read biographies and say, what balance of being running roughshod on people and being nurturing to people do I have to do to accomplish the mission that I've set out?
It seemed to me while reading that although she is a scientist and really enjoys pushing the frontier in the lab, she also does not shy away from taking on more leadership roles within the scientific community. Did she strike you as particularly ambitious and sort of resourceful as a scientist in terms of her ability to not only push the scientific frontier inside the lab, but also accomplish a lot outside of it?
I don't think that her accomplishments in the public policy and outside of the lab that you correctly referred to were driven by something so simple as ambition. I think she was driven by partly a moral compulsion. When she gets CRISPR and it's published and everybody's talking about it, she has a nightmare. And that's that she's invited into a room to meet somebody who wants to understand CRISPR, and the person looks up and it's Adolf Hitler. And she realizes deeply how CRISPR, the ability to design our own children, to edit their genes, to make them taller or blue eyed or do whatever genetic transformations we want to do in the human race or in our own family, that can be a force for good and it can be a force for evil. We've had enough experiments with eugenics in the 20th century to know how bad that can be. So she is driven to create a consortium of scientists around the world, in China, Europe, and the United States in particular, to figure out how far should we go with gene editing? Which are the rules of the road? So she has become a public policy thinker and leader, but I don't think she's doing it out of a desire to be Secretary of Health and Human Services or the World Health Organization or some drive for power or ambition. She just believes that, just as those of us who watched Oppenheimer know that scientists, once they create something new, have to sort of be part of the process of thinking through how do we use it, that she's unleashed something that is on a par with a nuclear energy. And she wants to be part of the process of saying, how do we use it wisely?
You mentioned some of the potential moral issues here. Can you quickly explain what happened in China in 2018 relating to CRISPR?
A Chinese scientist who had been to one of Jennifer Doudna's conferences decided to use CRISPR to make inheritable edits in the embryos or early stage fertilized eggs of what became twin children in China. The scientists used it to edit out the gene that allows HIV, the virus that causes AIDS, to infect the kid. Now, in some ways, that's a great thing. Their parents had HIV. If you can get rid of AIDS, that would be wonderful. But it caused an uproar because those edits were not only in those children, but they were in all the cells of those children, including their reproductive cells, so that none of their descendants would ever get AIDS again.
So, with that in mind, do you think that we have done enough in terms of creating the regulatory apparatus for gene editing since CRISPR was first discovered?
I think we've done pretty well in the United States. I know I seem like the optimist compared to some who may come on your podcast, but the Food and Drug Administration, they get it. People call them bureaucrats and think that they stand in the way of progress. No, the FDA has done a pretty good job of regulating the type of things you can do. The big problem, I think, is that it's got to be international. If you're not allowed to use CRISPR to edit your children not to get HIV or edit sickle cell out of them in the United States, somebody's going to set up a clinic in Bermuda, or they're going to do it in China or other places. So the challenge becomes, can you get worldwide buy-in, as we sometimes do on technologies, including nuclear weapons? We generally have buy-in. Can we have that with gene editing as well?
My view from reading the book was that it seems to me somewhat inevitable that CRISPR ends up in military hands unless some sort of regulatory body at the international level prevents that from happening. Would you agree with that?
Russia has not been a friend in this process. Putin seems enamored and says we can make soldiers who don't feel pain. We can. So, yeah, there's a problem if Russia goes rogue.
On that note, in terms of using CRISPR to edit ourselves for the better, you mentioned sickle cell anemia, which is a monogenic disorder, meaning it's the result of a variant in only one specific gene. There are other traits that are polygenic, where the output is a result of an interplay between could be 50 or 100 genes. So CRISPR right now can be sort of deployed to address a monogenic trait, and it's been deployed for sickle cell anemia. What still has to happen for us to be able to change polygenic traits effectively?
It's a very good question. So, obviously, those that are simple edits, a single gene, even a single letter change will do something. Those are easier to deal with, obviously. It's like a combination lock. If it's you only got to get one number right on the lock, you can undo it. But if there's 50 numbers you have to get in sequence, even a supercomputer has trouble unlocking 50 number combination lock. But the main thing is off target and unintentional at its unintended consequences. Even with the simple one of sickle cell anemia, and even with the simple one of a receptor for AIDS, making those edits might make you more susceptible to West Nile virus or something else. So we have to go slowly, step by step, to say what are the unintended consequences of fiddling with this gene or these 50 genes. And so it means that certain diseases or disabilities, such as sickle cell, Parkinson's, other things like that, will be easier. Muscular dystrophy could be easier. It's not a whole lot of genes. And those that will be the hardest are the ones that are so complicated we don't know the role of genes.
Right. You mentioned earlier the Human Genome Project. Do you think it's fair to say that the Human Genome Project didn't live up to the hype that it garnered at the time? And do you think CRISPR is one of the unlocks that will help it realize some of the potential that people thought it had at the time?
Yes, very good question. And we were all very excited. I was at Time Magazine. They're doing the Human Genome Project. We were writing about it every week. When they finally unlock it, we put Francis Collins and Eric Lander on the cover. Al Gore, who was vice president, said this will change everything. And then it didn't change everything. That's around 2000 or so. We pretty much get the sequencing. And for about 20 years, you've been around. Life hasn't been all that different simply because we sequence DNA. What unlocks it, to use your correct phrase in your question, is our ability to say, okay, now we can read it, but also now we can edit it. We can rewrite it. And that's what CRISPR and the use of RNA, the sister molecule to DNA, that's unlocks the fact that we've been given these scrolls that we can read helps us some in making medicines and drugs and pharmaceuticals. But once we can edit those information scrolls, that unlocks, I think, a whole new era.
The sentence that stuck with me the most in the book, and I'm paraphrasing a little bit, is something like, while kids of my generation have grown up learning the language of computer coding,
Mm-hmm.
kids of the next generation will grow up learning the language of genetic coding. Can you elaborate on that a little bit?
Yeah, I mean, I teach at Tulane, and sometimes parents will come in and say, I just want my kid to learn how to code. I don't want them to study Homer and Shakespeare or something. And I think, well, first of all, AI has come along. It's going to do coding a lot faster than your kid will ever be able to do it. What will count is the creativity and the morality and the ethics surrounding how all that happens. The coding that will be particularly important to decode is, how do you edit the human gene? Those questions you asked, which is, now that we've conquered sickle cell, and we kind of have, how do we conquer macular degeneracy in the eyes, or muscular dystrophy? And so learning how to do the four letters of the genetic code will be as important for the future as knowing how to do C++ or Python.
Walter, I want to be respectful of your time. I know we're coming up on time. Last question, unrelated to Codebreaker. Who or what topic are you thinking about focusing on next?
Marie Curie, for a lot of reasons. In fact, you caught me as I'm packing to go to Paris with my wife, Kathy, and my editor at Simon & Schuster. We're all going on a mission to go through her scientific papers. The big scientific breakthrough at the beginning of the 20th century was the understanding that chemistry is just basically physics. It's how electrons rotate around a nucleus and how they sometimes radiate and what radiation is. And Marie Curie, along with a few others, is the person who understands what 20th century chemistry, biochemistry, and physics are going to be. And she's also just a totally fascinating character. And her scientific notebooks, you can now analyze them using machine learning and AI, which is what I'm doing. I'm using something called Google Notebook, putting all of her scientific notebooks in and all of her papers and all of her letters, and analyzing exactly step by step how she makes her scientific breakthroughs.
Well, I look forward to reading the book when it comes out. Walter Isaacson, thank you so much for doing this. Thank you for writing a very digestible book on a complex topic here. And thanks for coming on to talk about it.
Andrew, it was a real pleasure, and it's a great pleasure to talk to somebody who's so prepared, so smart, and actually has read the book. Thank you, sir.